So maybe I can start off by welcoming you. Good afternoon. Welcome to today's Big Picture webinar. We have almost 40 people in the call. Today we have a webinar that will be provided by Gabrielle and Xavier from Work Package 5, and they will introduce themselves on the landscape of guidelines, standards, and regulatory requirements relevant for digital pathology in non-clinical setting. So it's a mouthful, but they will explain you all about it. Please keep the questions for after the presentation, and please raise your hand if you have a question. They will be ranked, and then all the way at the end we will go through them one by one. So I guess without further ado.
I think shall I share my slides now?
Not yet.
Not yet. Okay.
Yes, there you are.
Would you like me to share them now, or do you have something in addition?
No, please.
Okay.
You can.
Okay, so you should be able to see them now. This is our title slide, and Xavier and I will do this presentation in an alternating manner. Xavier, you are going to start, and I move the slide.
Okay, so welcome everyone. So if we go to the next slide, as a matter of a brief introduction, just for everyone to understand where we are in big picture. As you know, the consortium is divided into several work packages, and we are part of the regulatory work package. And a big chunk of the work package is about clinical, and we are the small part about non-clinical. And so in this context, there are a certain number of deliverables, and one of them was specific to understanding the regulatory background in the non-clinical area, which we have released already, but there were also some recent changes with a draft guidance from the FDA, so we've updated a little bit the information that we're going to share today. And ultimately, what we want is to build the relationship with the authorities to help get acceptance, not only for the GLP use of whole slide images, but also to introduce AI models in the future. So in terms of the presenters today, both Gabrielle and I have been working in the field of toxicologic pathology. Gabrielle brings more than 30 years of experience in the field. She's worked for different corporations. She's had her own consulting business as well. I'm bringing 20 years and also working for several corporations, including CRO. And so at the moment, Gabrielle is leading the pathology group at Boehringer Ingelheim in Germany, and I'm leading the pathology group at Pfizer in the US. So with that, I think we can move and restart the presentation.
Okay. Yeah, I'm presenting to you now the table of contents. What are we going to tell you today? First of all, we will have a very short introduction about what are non-clinical safety studies and what is our work in toxicologic pathology. It will really be brief because only a couple of weeks ago we had a nice webinar given by Julie Boisclair and Brian Knight more extensively about toxicologic pathology. We will then mention what is GLP, good laboratory practice. And under 3, you can see a couple of points which touch on aspects that are important for our work in toxicologic pathology, where we will present in an alternating mode. So I will always present what is the conventional understanding of GLP for these aspects, and then Xavier will tell you what are now the challenges for the future with digital pathology. And finally, we will talk about the next steps in our endeavor. So in non-clinical safety and in toxicologic pathology, what we do is that we identify hazards for humans and characterize those hazards by evaluating a biological test system which is not man. So this is laboratory animals. In those studies, among many parameters, we are doing pathology evaluations. So among others, there are analysis of body fluids like hematology, coagulation, clinical chemistry, urinalysis, evaluation of bone marrow. And then at the end of a study, all animals are killed and necropsied. We evaluate grossly the organ morphology. We take organ weights. And finally, we do an extensive histopathological evaluation of all animals. There can also be special investigations in such studies, like special stains in histopathology, evaluation of biomarkers, immunohistochemistry, and others. It's important that we do an integrative assessment, so we do not do an assessment in isolation, but we have to look which are the other study results and which are correlations between findings we have. The overall goal of such a non-clinical safety study is to determine target organs of toxicity in that given species and to assess the relevance of these findings, because they are used in order to determine a no observed adverse effect level, so a dosage in those non-clinical studies, which is then used with a calculation, with a formula to set the dosage for first studies that are run in humans. But we are not only looking at these animals, we are asked to investigate toxicological mechanisms leading to those changes, to assess reversibility of the findings, and to look at the translatability. What is the relevance of what we see in an animal for humans? Can that also be seen in humans, or is it something species specific? We also look at the monitorability of findings. So in the first trials in humans, do we have a possibility, do we have means to find indicators for such changes very, very early to, in addition, be able to prevent humans from any uh, noxious effects. And finally, we also have to bear in mind what will be the patient population that finally will be treated with such a drug, because that also influences our view on changes which we can see. Very briefly so, our studies are ruled by international guidelines. So there are laws which tell us which types of studies have to be conducted for which type of use in humans. Generally, studies have to be performed in one rodent species and one non-rodent species. Non-rodent species can be beagle dog or mini pig or non-human primate, according to which species suits best for a given compound. Usually, such studies contain one control group that is treated with the vehicle only and 3 treated groups with increasing dosages. Then we have to perform studies of increasing treatment duration depending on the intended clinical use in patients. And as I said before, among numerous other study endpoints in pathology, we look grossly at organ morphology, at organ weights, and do extensive histopathology. To give you an impression about the dimensions or the numbers of tissues we have listed here some typical studies performed for new drugs. A 4-week toxicity study, so treatment for 4 weeks in rodents, so either in rats or in mice. They typically contain 10 males and 10 females per group, killed at the end of treatment, and then also recovery groups kept for an additional period in order to evaluate reversibility of changes. And just to give you an impression for that type of study, for pathology, we end up with 120 animals in such a study. Here in gray, you can see study groups which are not evaluated in pathology. They are used for other study endpoints. A longer duration would be a 13-week toxicity study. So here you can see the number of animals at the end of the study for pathology is higher, would be typically 160. And a carcinogenicity study, which is lifespan treatment of rodents for 2 years, with 50 males and 50 females per group, so that we end up with at least 400 animals in such a study. And this is also important to give you an impression about the dimension. Here you can see a slide tray with something like 25, 26 slides per animal, a rat here, and all the organs contained on these slides. So you can see that on the one hand, for some organs, like here is the brain, we have several sections per organ. But on the other hand, there are small organs, like these are different intestinal segments, which can be grouped together on one slide. Altogether, this makes something like 50 organs per animal, and we produce something like 1,000 slides for a 4-week study and have to evaluate more than 2,000 tissues in such a study. So I'm now coming to GLP. What is GLP? GLP, good laboratory practice, concerns the conduct of non-clinical safety studies, especially those which are used in order to extrapolate a risk to humans. Those principles concern the planning, the performance, monitoring, recording, reporting, and archival of those studies. And they were put into place in order to assure the regulatory authorities that data submitted in order to obtain the authorization to go to the market with new drugs can be relied upon. So they should ensure the uniformity, the consistency, the reliability, reproducibility, quality, and integrity of the data presented to authorities. They are therefore an absolute requirement for market authorization. And test facilities performing such non-clinical studies are regularly inspected by national GLP bodies. So in Germany, it would be the German national GLP body. And in other countries, they are national bodies, but there is mutual acceptance of GLP compliance between the OECD member countries. And it must be said that everywhere in the world, in the past and in the present, and very probably in the future, the regulatory body in the USA, so the FDA, is really something like a guidance for all countries. Okay, and now we are coming to the various aspects of GLP which are important for toxicological pathology. The first aspect we identified is around specimens and raw data. Specimens are material derived from a test system, so in our case from a laboratory animal, and that are then examined or analyzed and have to be archived. In our case, that would be the wet tissue, so organs preserved at necropsy and fixed in formalin, paraffin blocks, and the glass slides produced from them. So this is for the traditional toxicologic pathology. Raw data are defined to be worksheets or records or notes that are the results of observations and the results of a study, and which are necessary to reconstruct a study, so to understand how a study was conducted and how results were obtained. It must be said that exact copies traditionally are accepted to replace the original raw data, but they have to be dated and verified accurate by signature by the person taking the copy. In traditional toxicological pathology, raw data can also include photographs, computer printouts, and recorded data from automated instruments. And then a specificity for histopathology is that raw data are not considered to be notes taken or records immediately made at observation, but only the finalized and signed pathology report, because it is recognized that when evaluating, as I showed, something like 2,000 organs of a study, there is some back and forth. There are some thoughts, some consulting textbooks, some literature research in between, some consultation with colleagues before one is really sure how to call changes and how to evaluate relevance of changes and how to judge differences between treated animals and control animals. So for histopathology, raw data only begin to exist with the signature of the pathology report. And then very important, specimens, if they are not perishable like blood smears, but all other specimens and raw data have to be archived. Now the next slide.
So, as you understood from Gabriel's presentation about raw data and specimens, the problem we have with whole slide images when we use them in order to derive diagnoses and then to generate raw data that will be used to reconstruct the study report is that these whole slide images actually are not real specimens because they are not directly derived from the animals or the test system, and they are not the dated and signed pathology report, which is considered the raw data. So they sit in between, and there's been a consensus. There's no regulatory definition for that so far, but there's been a consensus, and the concept of a faithful replica to identify and characterize these whole slide images as neither specimens nor raw data has been put forward. So that's the first thing, and we hope to get regulatory consensus on this definition. Then the other important thing is that when we use the whole slide images in this specific context, um, we think we and what's been published and what's been put in the regulatory guidances so far is that we will have to archive these whole slide images. The duration of archiving, again, is not specifically precise in the guidances, but the recommendation is that that should follow the rules for specimens. And the rules for specimens, they depend on local jurisdictions, and also they might depend on top of it on specific company policies. So it may be country specific. And then the last thing, because of the digital nature of whole slide images, there is a bunch of regulatory recommendations in the context of electronic records and electronic signatures that will have to be considered and that will have to be followed for the whole slide images. Next one.
Yeah, the next aspect we would like to present is about edits or modifications of raw data obtained in a study, and this then also touches on data integrity. So yesterday and today in toxicologic pathology, it is stated that specimens must be identified to confirm their origin and enable their traceability. All data generated should be recorded directly, promptly, accurately, and legibly, and must be signed or initialed and dated. And then any change in the raw data must be made so that the previous entry can still be read, can still be seen, must indicate the reason for change, and must also be signed and dated. So change in the raw data then will also be important for the new aspects of digital pathology. Data generated as a computer input must also be identified at the time of data input and by the individual doing it. And finally, computerized systems must retain full audit trails in order also to be able to show the original data and all changes made, together with timed and dated signatures. And again, those reasons for changes.
So regarding whole slide images, this principle of data integrity is also a key component when we'll be inspected using these whole slide images in a GLP context. This is precisely what the inspectors are going to dig into. They will want to make sure that we can reassure the public who trusts us to test our compounds that we have preserved the data integrity all along our evaluation. So in general, the part of regulation in the US that's about electronic records is part of the CFR, the Code of Federal Regulation. It's 25 CFR Part 11, and it gives a certain number of recommendations regarding the manipulation of electronic records. So in particular, the raw data need to be preserved for integrity, and they can only be modified if there is an audit trail, which is a record that somebody has modified the raw data at a certain moment in time, and also timestamps to know exactly when the raw data has been recorded, because it has to be recorded in a timely manner. You do not record the raw data 2 days after the facts. Similarly, compression is under scrutiny because you don't want to lose information. So if you are to compress something, you'll have to demonstrate that you are not altering the original record. And as I mentioned already, you need to record in a timely manner. You don't want to have a gap between the generation and the recording of your data. So in terms of the readability of the data over time, again, there is nothing here in the regulation, but the consensus across industry partners is that for long-term readability, and we'll talk about that when we'll talk about archiving, and to get away from proprietary formats, a tendency, a trend is exactly as we are suggesting in the Bigpicture Consortium, is to use a format like DICOM, which will be prone to containerization of metadata, but also prone to readability over time. So this is where we are going at the moment. But again, regarding this format, there is no regulatory guidance. Then we can go to the next slide.
Yeah, now we are coming to pathology peer review, and that will take a little bit longer to explain. So we have to distinguish between the initial slide evaluation by the study pathologist and then a pathology peer review, which is done by a second independent pathologist. This is more than just an informal consultation with your colleague sitting next door. It is not an absolute requirement by guidelines, but it is true that it is somehow expected nowadays from all regulatory agencies to be performed. As I said, a second independent pathologist will do this peer review, not in a way that he or she will produce de novo diagnosis, but will try to understand the way the first pathologist saw changes and described changes by reviewing a subset of the slides and the draft report. Usually, this peer review is performed before the pathology report is finalized. So if you remember right, that is before raw data are created. It will contain a discussion, and then in most cases will end up with a consensus, so a common understanding and agreement between both pathologists. And in that case, it is called contemporaneous because it is done during the pathology evaluation and before creation of raw data. A pathology peer review can also be done in a more formal way after the finalization of the pathology report. Then it is called retrospective. That needs more documentation because raw data already exist with the signature of the pathology report and are modified with this peer review. And finally, if those both pathologists do not achieve consensus, an even more formal process, a peer review working group, so a group of experts can be installed with very formal instructions how they evaluate the slides and come up with a common or individual opinion. We have a diagram showing what this pathology peer review is like. So we have a contemporaneous peer review on the one hand, which would be conducted when the study pathologist has completed his assessment but has not yet signed his report, or a retrospective peer review when the pathology report is already signed, so raw data exist. The peer review is a review of a subset of slides review of the draft pathology report or signed pathology report, review also of other pathology results, and of course needs a lot of background knowledge and understanding of the study, understanding of the compound, and so on. So slide reading of a subset of slides will be done, review of documents will be done, comments and differences are discussed between the peer review and the study pathologist, and a consensus is achieved in most cases. If a consensus is achieved, a peer review statement is signed, and now traditionally the pathology report can be finalized and raw data are only created then. That doesn't need any further documentation than the peer review statement. However, if it was a retrospective peer review, so raw data already existed, then that peer review contributed to the creation of new raw data, and the report will be amended even if only minor changes have been done. That is important to understand for what Xavier will now talk about, what it means for digital pathology.
So as I mentioned before, a draft guidance was issued a couple of weeks ago from the FDA on the topic. And when you look closely at the guidance, these are question and answers. There's one question about what needs to be retained when we're using whole slide images. And the answer needs to be clarified because it's not exactly what we think it would be. And in particular, when you look at the answer, it says, without any distinction between contemporaneous and retrospective peer reviews, that the whole slide images should be archived. And that's where we need to get clarification from the authorities, because our perspective is that we need to make a differentiation between contemporaneous and retrospective peer reviews for the exact reason that Gabrielle mentioned, which is in one case, the raw data has not been created yet when it's contemporaneous. In the other case, when it's retrospective, the raw data has been created and then we are modifying the raw data. So in the first case, contemporaneous, considering that the primary read has been performed on the glass slides, and then we're going to derive the diagnosis from the glass slides, we're going to keep the glass slides, and the raw data will be generated after we have consensus after the end of the peer review. We think that the whole slide images are have a comparable status to notes that we are using during the peer review. They are a tool to help the peer review, to help with the discussions during the peer review, but they shouldn't be kept and archived. So that's a big thing that needs to be clarified, and the draft guidance is now has been submitted for comments, and we know that a lot of these comments are around this specific topic. And then it's easier for retrospective peer reviews and for primary reads. We agree that in that case, because the whole slide images are used to derive diagnoses that will change or either generate or then change raw data, then there is a need for archival, exactly as we archive the specimens. So we can move to the next one.
Yeah, the next one is about multi-site studies and material transport, because that is also very important when we will use digital pathology. So you already noted that there are two pathologists working on a study, the study pathologist and the peer review pathologist. So you can already understand that there will be some transport between these two pathologists. They can be at the same site, at the same location, but also at more than one site. A multi-site study is a study that has phases, so let's say activities conducted at more than one site. Imagine there is a study with an in-life phase conducted at a test facility, but then slides can be produced in an external lab. From there, slides can be sent back to the test facility for a pathologist to evaluate them. And from there, slides can be sent to a peer review pathologist at another site for peer review. So there can be a lot of transports involved, even for glass slides. For traditional toxicologic pathology, as I said, transports can be wet tissue to a contract histolab, slides from a contract histolab to the sponsor or to a contract pathologist, or slides to a contract pathologist, slides from a contract pathologist or from a contract research organization to the sponsor for peer review, and then remaining wet tissue, paraffin blocks, and slides can be sent to a contract archive or back to the test facility for archiving. For transfers and transports, mechanisms to maintain data or material integrity need to be in place. That means a secure transport has to be ensured, and there needs to be a chain of custody documentation in order to prove that what has been dispatched has also arrived and been taken care of. We have a diagram with several possibilities of sending material around, and we found that this is important to show to you because it also shows here the advantage that we will have once we can work digitally. Like, for example, here you have a pharma company which is producing the histo slides, is then scanning slides, is handing them over to a pathologist for primary evaluation. Then they go to a second pathologist for peer review, and finally the slides are archived. But we can also have the scenario where slides are produced and scanned and are sent to an external contract pathologist for primary evaluation, and from there are sent back for peer review by a sponsor pathologist and finally are archived. Or let's be more complicated. We can produce slides at an external contract histology lab. They would scan the slides, or they would send the glass slides and they would be scanned back at the sponsor facility and then go to primary evaluation, peer review, and so forth. Or we can have a study performed at a contract research organization who would produce slides, scan them, ensure the primary pathology evaluation, but would then send the glass slides to the sponsor pathologist for peer review. And from there, they are sent back to the CRO for archiving. And finally, there could also be the slide scanning that be done by the sponsor's test facility before doing the peer review and sending back slides to archiving. And these are not all possible scenarios, but you understand that there is a lot of transfer and a lot of transport within these scenarios, and that sending slides digitally will be a big advantage.
And so we are back to data integrity in the context of transferring digital slides. These are the same expectations as when we are transferring glass slides. So in that case, not only we need to transfer the whole slide images, but also the metadata that accompany the images. And the expectation remains the same. We don't want to lose anything on the way. So there are several mechanisms that have been proposed, in particular performing checksum comparisons, which is basically a comparison of the amount of material at the beginning and the amount of material at the end. There are different ways to do it. This is the proposal that's been made so far, but there is no regulatory guidance to confirm that this is okay. It's an acceptable way to reassure in terms of data integrity. Similarly, what's been proposed was to incorporate one tissue or one slide with multiple tissues, could be a tissue macroarray slide, for the purpose of QCing. So you would send your digital images, and in parallel, you would send a physical slide for the purpose of QCing. And so the recipient would, of course, look at the digital slides, and for this given reference slide would check that what they see in terms of digital slide is what they see under the microscope for this one. That was an original proposal that's been debated because the current thinking is that it's not necessarily needed as long as the validation, and we'll talk about the validation, has proven that they had no issue in terms of data integrity and the transfer was trustable. So again, there is no guidance here. It's for discussion, but these are the two options that have been proposed. Regarding the draft guidance that's been published, so you can see pretty similar principles. If we are to use whole slide images for GLPUs, the expectation from the FDA is that we're going to have a backup file. So you could see that as something parallel to having blocks when we are using slides. There's a way to get back if needed. A chain of custody, exactly as we have for specimens. And then we have to control accesses, who is able to access to this data, how the systems are secured, how the transmission is performed, and then make sure we have procedures and documentation around that. And as I mentioned before, this falls under the umbrella of 21 CFR Part 11, and the whole principle is really to reassure regulators that we maintain the data integrity. So now we're going to move up to qualification and validation.
Yes, qualification and validation. Let me present to you what is said in the guidelines about qualification and validation of equipment traditionally. It's the responsibility of test facility management to ensure the quality and fitness for use of equipment and materials, and that acceptability of equipment and materials has to be demonstrated to regulatory authorities. So a lot of documentation that has to be shown. For computer systems and softwares, all computer software should be acceptance tested before being put into service. Then a full documentation of the validation process and acceptance tests must be available and should be retained in the archives. And usually the very first steps before validating equipment are to establish acceptance criteria. So a lot of brain work thinking about what do we expect, what is the intended use, and what are criteria in order to state about fitness for use of an equipment, and to perform a vendor audit. So go see the vendor of an equipment in order to have a thorough idea about how they work, how they establish the fitness of use of their equipment for their clients.
So regarding the digital component, we'll start by highlighting differences between the clinical world and the non-clinical world. And then we'll go a bit deeper in the scope of the validation. And then we'll talk about SOPs. So in terms of qualification and validation, there is a big difference between the clinical world and the non-clinical. Things are pretty well defined in the clinical world. There's an FDA guidance. There are recommendations from professional societies that people can rely on. And one of them is to qualify the scanners as medical devices. It's been done. The first one, the Philips IntelliSight, has been qualified a few years ago. So people have a very good reference. It's not the case for non-clinical. And on top of that, the context of use is different because what we do is really a comparison between controls and treated animals. And we're not looking at making diagnoses as we would be looking to for humans. So this is an important component because a lot of the slides that we're going to evaluate are for a different purpose. And a lot of them will be completely normal, by the way. So, so far, in terms of expectations for the qualification, this is true that the current draft guidance is well aligned, except for a few things with the recommendations from professional societies. And there is no mandate for the scanners to be cleared as medical devices. It seems to be acceptable to take the same route, but this is not a recommendation. And in practice, most companies are using scanners that are not the ones that are used for clinical use. So we can go to the next one and dig a bit deeper into the scope of the validation. So the framework is the same as what I've explained before in terms of 21 CFR Part 11, which is about electronic records, and also 58, which is the general principles of GLP. So we have to make sure we stay under these big umbrellas. So when you look in details about what is needed, and this is coming mainly from publications from professional societies, uh, what we expect is to have a preliminary step of a vendor assessment, and there are a certain number of things that we are supposed to verify. In particular, if the vendor has a quality management system and how they document what they are doing. So in particular, what is the access looking like? Who has access? Who is the user? How the roles are defined? Again, the thought behind is to make sure that we have no unauthorized users who might be in a position to modify the data. What are the coding standards? What about the security? What about the penetration to the system? Are people trained properly? Training is one of the requirements for GLP. Um, what about the testing? What about the release? What about the change control? What about the version control? How do we manage problems and how do we solve issues? So all this preliminary evaluation is what is expected as part of the validation report. Then the scope of the validation has been referred to as the pixel pathway. The idea is really to follow from the generation of the virtual image up to the end of it, including archival. And we should make sure that there is no loss of information all along the image chain. In terms of the scanner itself, so there are technical parameters that can be taken into consideration. And honestly, we copy some of what's been done in terms of the clearance of the scanners for medical use. And again, the big thing is to make sure that we maintain integrity, in particular when we exchange files or when we modify the format of a file. And we'll talk about that in terms of archiving and our position on that specific item. And then similarly, there's an expectation that the people who are using the system are going to be trained, so the pathologist in that scenario, and they'll be confident that the whole slide images are a faithful representation of what they would be observing on the microscope. There are studies that can come as part of the validation package. They are called non-inferiority studies, and so the purpose is to demonstrate that evaluation through whole slide images is at least as good as what we would do from glass slides. There's also a difference here between these non-inferiority studies or concordance studies between clinical use and non-clinical. In our case, it's probably much more appropriate to mirror or to mimic the evaluation of a study rather than to put together an extensive list of diagnoses and tissues as what would be preferred in the context of a clinical use. And there's this notion of example slide that I have mentioned before, which is under discussion. So the next slide is more about the need for SOPs. So in the principles of GLP, we have to describe what we are going to do through SOPs, through study plan, for example, and then we have to execute according to what we have described that we would be doing. So these are the basic principles. And so there is no exception for the use of whole slide images. So these SOPs will be applicable to people involved throughout the whole process, starting at scanning, up to evaluation, and up to archiving. And each individual step in the workflow needs to be described in a standard operating procedure. And as I mentioned before, there's a need for people to be trained. That's really part of the basic expectations. So when you look also at the retrieving of information, and we will go into more details when we talk about archiving, there is also an expectation that the retrieving of information and archiving in general is covered by a standard operating procedure. So we can go to the next one.
Yes, now we're coming to archiving. So archiving of materials associated with non-clinical safety studies. What is said in the guidelines traditionally? Archive facilities should be provided for a secure storage and retrieval of study plans, raw data, final reports, samples of test items, and specimens. The archive conditions must protect contents from untimely deterioration. Archiving may be outsourced, but in that case, there needs to be an access arrangement for the depositor and also for inspectors. There are obligatory archiving periods which are not defined in the OECD documents, so they are not internationally harmonized, but they are governed by national laws. Like, for example, in Germany, there is the Chemicals Act, which requires 15 years of archiving after study finalization. In reality, in very, very many cases, materials are even archived throughout the, let's say, life of a drug. The traditional understanding of an archive for inspectors is that it is a physical archive. It is a locked room that can be inspected. An archived material is then accessible and can be controlled for completeness. Because this here is said, the archive and retained materials and records should be available for inspection during normal office hours. If electronic records are kept, it should be possible to make them available in human readable form. So this is something which now we will have to translate to what it can mean in the future. Computerized archive facility must have processes to prevent unauthorized access. And finally, again, the format of electronic records must be readable for the duration of the archiving period, which can be a true challenge.
So if we try to transpose these principles for digital pathology, the first thing to say is to understand that yes, it is okay to migrate the data for storage. And so usually the storage and archiving is segmented into three parts. There's short-term storage, there's mid-term storage, and there's archival. There are different manners for storage to happen. It can be a physical separation, same computer, but outside of the production area. It can be a logical separation, so there's a notification that the material is archived, but there's also a need to lock this material. In terms of archiving, the expectation is that the data are on a separate and dedicated archiving system. And it's not only the images, it's the metadata, it's the audit trails, it's the electronic signature, and it may also be hardware and software, in particular if the record medium requires processing in order to be able to read the data. If you were to archive on magnetic bands, for example, then you'll have to have the reading system so that at the end of your archiving period, and if you have an inspection, the inspector will be able to get back to these magnetic bands and have a look and really get the data. So it's something to consider because also in terms of the format you want to archive, you want to make sure that in certain number of years, you'll be able to read the format and you will have the hardware to get access to that. So if you look at the draft guidance that's been released by the FDA, it's interesting that doesn't speak much about archiving. And is that intentional? I don't know. What is sure is that there is an open question because, as Gabriel mentioned, that archives should be amenable to a physical inspection. The question is open for electronic archives, in particular about the location of the servers. Do we expect, or do the regulators expect, that the servers will be on site, on premise, so that the day of the inspection, they can open the door, they can check security, who has access and whatever? Or is that acceptable that the servers are not on site and not necessarily directly amenable to a physical inspection? And so this remains to be clarified. If you go and look at the OECD document, and there's an FAQ on the topic, it just gives general principles there. And in particular, it's again around the notion of integrity and the notion of accessibility of the archives over time. You can go to the next one. Going into a bit more details about OECD expectations. So this is the guidance 17, which is referenced here. And that says a certain number of interesting things. First, yes, it's okay to use electronic archival for any GLP relevant data. So we can archive whole slide images. But the expectation is that we'll have similar access, similar levels of reassurance in terms of controlling the access, indexing the data, and being able to retrieve the data. There's also a strong emphasis on the notion of data integrity. So we don't want this archive to be amenable to alteration, deletion, or replication. But there's a distinction that's been made about the access to the data. You can see that as for physical archives, you can go into the archives and you can consult a document and then just leave the document in the perimeter or archives and just have a consultation and then give it back. Or you can retrieve the document. You go to the archives, you get the file, then you bring it to your desk, you read it, and potentially you have risks of losing something or altering something, and then you bring it back to the archives. And so there's a similarity which is mentioned here in the OECD guidance under paragraph 111, which says that if you just look at the data, you do not necessarily perform a retrieval of the data. So we need to define the rights of who has access and what happens to the data. And there's an expectation that a dedicated archivist, exactly like for specimens, will be in charge of it. So this is something that we need to take into account pretty upstream when we're thinking about the validation and the infrastructure where we're going to keep our data. There's a need for validation. As we mentioned before, archiving is an independent process. So we need to be very careful that we're not going to validate the use of whole slide images and then just forget this component, because that's one of the key components as well. And because it's under GLP, it has to be covered and described by SOPs. So there's nothing very new here. And at the end of the day, if we are to use whole slide images and then we archive them, this has to be put black on white in the study report. The study director at the end of the day has to say, yeah, we have used whole slide images, they have been archived, this is the location. And the SOPs will tell us for how long the data are going to be archived. So in general, the expectation will be that these whole slide images will follow same durations as specimens. And so again, national jurisdiction, company policies. In my case, that would be 30 years. So if I'm to archive whole slide images, I want to be sure that in 30 years from now, there is an inspection at Pfizer. Hopefully I'm retired, but someone will be able to read and have access to the whole data without any alteration. So this is the expectation. Okay, next slide.
And the next slide is the last slide.
So what are the next steps? So we are in touch with the regulatory authorities. We've put together 2 meetings, one of them coming up next week with the FDA, and the next one, the following one with the EMA, will be early September. And so the idea is to foster the communication and bring put on the table some of the things that are not necessarily clear, or the things that we would like to get reassurance, because many companies are validating or even have validated. There's been an announcement from Charles River that they had validated the whole workflow. And so there's a notion of risk taking here, because as we do see consensus emerging here and there are a few gray areas. And so maintaining the discussion with FDA, coming with our proposals and see how they respond to that. Of course, they are not going to put a strong commitment during such a meeting, but at least for us being able to feel if we are really in the wrong direction or if they are amenable to it is the first thing. And also because there are gaps in the current draft guidance on whole slide images in GLPUs, we'd like to suggest that they expand the guidance and make it more exhaustive in order to cover our needs. So with that, I think we can conclude and open up for Q&As.
Okay.
I think I will stop the presentation because otherwise we don't see what is going on.
Thank you very much, Xavier and Gabriele. I think it was very insightful. I see some hands raised. We don't have very much time, so we can take a few, and then I'm sure Xavier and Gabi will be reachable by email.
Absolutely.
Okay, so first one is Pierre. Raised his hand. Please
Yes.
unmute. Yes.
Can you hear me?
Yes.
Yes.
I'm not sure I was the first one, but I will be quick. Thank you very much for the kind presentation. Just a few remarks from my side. For the compression of the image alteration, I'm not completely convinced this is a key parameter to consider. I would rather focus on the slide equivalency between the physical and the digital slide. To allow a proper evaluation of the scan versus the physical. And I think also every company using a scanning and a digital image should also evaluate the limitation of the system, because we know that some slides we cannot evaluate with the scans, like with birefringent material, etc. So there are some clear limitations have to be evaluated and pondered, let's say, before starting or some tox studies. Yeah. Otherwise, thank you.
Yeah, that's a good point, Pierre. This is about the context of use. And there's another point that we have not necessarily addressed here is how do we manage the intermediate file formats in terms of archiving in particular. And the thought here, or the predominant thought here, is that we don't necessarily need to archive all intermediate formats. So starting from the format that's generated by the scanner, and then the proprietary format that might be generated for the viewing of the images, and then the archiving format. Again, if the validation has the burden to demonstrate that there is a faithful replica at the level of the scanner, and then there are true copies in the subsequent steps. Then if we are able to demonstrate that, then we can probably make a case for not archiving the different formats, which is very important in terms of space, because if you have a triplicate of your files, in terms of the space it takes, it's very detrimental.
So just a point of order. So it is 4 o'clock. I think, I don't know, Xavier, Gabi, if possible, we could stay a few minutes longer for to discuss. But then people who have another meeting can run away. It will be recorded, so the Q&A, you can also follow it. You can look at it on the SharePoint. But first question is if Gabrielle and Xavier are available.
Yeah, for me it's possible.
Yeah, same here.
Okay, perfect. Then for the people that have to leave, thanks a lot for your interest. And here we will continue the discussion. The next one was Peter Hall.
Hi, Gabby and Xavier. Thanks for your presentation. It's very helpful. So I've got a couple of questions, and I'm not sure you can answer them necessarily, but you talked about peer review being an expectation rather than a
You're breaking up,
Oh.
Peter.
Hello, sorry, can you?
You're still breaking up.
Yeah, so maybe you can turn off your camera. That may help. But
Yeah, okay. Yeah, so I'm not sure whether you can necessarily answer this question, but it's something which kind of plays in my mind a bit. So you talked about peer review as a expectation rather than a requirement. But what do you think about having a peer review which is non-GLP compliant on a GLP study? Do you have any kind of thoughts on how the authorities would, this is for digital peer review, how the authorities would regard that if you've gone through the process maybe of ensuring the pixel chain is faithful, but you haven't gone through the longer process of validating the system? Do you think do you think that would be, how do you think the regulators might respond to that?
So I can start, and you feel free to chime in, Gabby. I think it's a question of reassurance. There is no expectation that peer review has to be GLP, but of course, if you claim for GLP compliance, you have to follow the rules. So you can still have a peer review and have an exception to the GLP. That's going to be part of your report. So it's technically doable, but and so it becomes a personal thought. It's just my own opinion. If the purpose of a peer review is to increase the confidence that a reviewer will have when reviewing your study report, um, I mean, I would tend to favor a GLP peer review, because that's also part of the GLP to reassure that we have, paid particular attention to the processes in terms of integrity and so on and so forth. So to me, it's like adding two steps that converge to reassure a potential reviewer.
Yeah, I completely agree, Xavier. Although I would like to add that there is such a pressure now in the pharmaceutical industry to be able to use digital pathology, at least for peer reviews, that I believe that if there is no further practical guidance in time, that there may be companies who switch to non-GLP digital peer reviews because they really need to do that. Yeah.
That's the path of least resistance. I agree, because the validation is a huge endeavor. So if you don't have the resource to do it, I mean, there's a temptation to say, yeah, okay, we're going to use the whole slide images, but non-GLP.
Yeah, and can I ask a little follow-up question then? If you're just going to go down the peer review route, and you have your slides scanned, say at a CRO who's GLP compliant, and you use a system that's well known and has been validated by other people, how much effort is involved in actually doing your side of the validation process? So the actual, from receiving the slides to doing the peer review, is that, presumably that's less intensive than the whole thing, but can you give me an idea of the effort or the resource required for that?
That's a whole workshop on its own.
I know, I'm sure. Sorry, but you can see where we're coming, where we are with that kind of question, which we're in that position where we're thinking about it and, trying to figure out how much we need to invest.
So you would have to validate for sure the transfer of data, making sure that there is no loss of integrity from the time the CRO has let the data transfer to your site. And then you of course you still have to validate your viewer software, and up to then you have to decide what you're going to do with the data, how you're going to manage the archiving process. So it saves maybe a little bit, but I'm not even sure, because what you save in that case is the validation of the scanner, but you'll have to validate the transfer of the data anyway.
And what would you be archiving out of interest? Because, you'd be making notes, presumably from the digital slides, and then discussing that with the study pathologist?
So, so there's a notion here that in the context of the peer review, if you're making notes as overlay on the image, or like a pinpoint or something like that to help for the peer review, then you should not necessarily consider that as an alteration of the image. And so similarly, if you don't keep the images in the context of the evaluation of a primary on the primary evaluation on the glass slide, for example, so these annotations would not be considered as altering anything, and you would not have to archive your whole slide images.
Okay, thank you for that.
That'd be like exactly like notes that you would put on an incidence table, for example, or on the printout of individual animals, highlighting the findings you want to talk or discuss with the pathologist at the CRO. So that would mirror this practice.
Okay, thank you.
Okay, so there's one or actually two more questions. One from Moritz, if he's still around.
Sure. So I was just wondering, the DICOM format we're proposing for Bigpicture, are we going to use for Bigpicture? Is that, did anybody, or is that an option you would consider as like a standardization for archiving? Or is there? So I guess there's multiple questions. Would there be a benefit to having like a standardized common archiving format? One, B, is it feasible? And C, is it would it cause more trouble to check for the integrity of the information during that transformation? I would assume that just using the format you scan with is the best thing to archive, because that's, you know, the original. But would it make sense to kind of go for standardization with that?
So
I
then
think the
you go ahead.
Yeah, I think the fear is that if the format of a scanner is used, that is a proprietary format, and long-term readability is more difficult to ensure than if you use the DICOM format. But as you say, Moritz, of course, that then includes an additional step for validation or for proving that there is still the notion of faithful replica up to the DICOM format of a whole slide image. So my understanding as a pure user is that using the DICOM format for archiving somehow is a wishful thinking, because that would facilitate long-term archiving, but it has not been achieved. I'm not aware of any regulatory answer or something concerning a format.
So for now, let's wait.
What do you think, Xavier?
Yeah, the advantage of DICOM is that it's followed over time. There are specifications that are made available. It's been used for the archiving of other medical records, imaging in particular. So mimicking that use would make perfect sense. And as you mentioned, you get away from proprietary formats. So for long-term retrievability of the data, that'd be the preferred option.
Okay, so if there's no more questions, I would really like to thank Xavier and Gabrielle. It was at least for me, it was very insightful, and also looking at the discussion, I think there was a lot of people are interested and like to think along with all the challenges that are faced in transforming to digital pathology in a GLP setting. So thanks a lot for this webinar. Just one more thing I want to mention is that the next 2 months, so July and August, we won't have any webinars. And then the next one will be on the 16th of September on explainable AI. which is also a very interesting topic, I think. Moritz, yes, you can.
Just a quick one, sorry, if there's
Yes.
a few more seconds. We've been discussing whether in our electronic system where our slides reside for primary reading, not for archiving, whether it's okay to, similar to what you said, drawing errors for peer review or something, whether we can, because they're just add like a free text field where the peer reviewer can put notes in, like, for example, yes or no, or disagree, or I think this is that and this. And my personal opinion would be yes, because it's not going to be archived. It's just some form of note. It would be like an Excel sheet or some Word document or an email, but it's in there. So there's a lot of concerns from some sides that this would be then considered some sort of raw data and then be a big problem because it's not secure. It's not all kind of things. Did you talk about this before with anybody?
Yeah,
Or
so
something?
personally, I would consider that as an overlay as well. And if you try to mirror what you do with slides, I know how many of us, when we're looking at slides, we take a marker pen and then we put something on the slide to direct the evaluation to that specific area. So I see that as something pretty similar to it. And then at the end of the peer review, you clean your slides and it kind of disappears. Yeah.
But I'm not sure if I understand, Moritz, because you say that is not archived. So where is it recorded then? I understood that it's kept together with the digitized slide.
So we haven't figured out how we want to do the archiving of the digital slides yet. So this is slightly foggy. But at the moment we're using a digital system Concentric for storing and viewing these slides, for working with them, and it has options to add metadata fields, and you can also add a free text thing there, which could be altered by the peer reviewing pathologist, and you could just write in there whatever you want, or make it more complicated, or have like tick fields or whatever. And so it would be with the slide in the storage where we would actually use the slides. And now the next question is, where is our archive going to be? Can we lock the slides in there so that this simultaneously could be considered an archive? But unfortunately, we think it cannot be at the moment because it's not unalterable enough and not audit trailed enough. It's audit trailed, but maybe not enough. So we would probably have to put the slides into a separate archive, we think, and then those comments would be with the slides.
So in fact, I would never put such notes into an archive because, as I said about the contemporaneous peer review, it's really the common understanding that you achieve a consensus at the end of the process and that you do not create new raw data. Now, if you have those notes, yes and no, they will show some disagreement to an inspector that you do not want to show, because at the end you came to a consensus. I would certainly not take the risk to archive such notes.
Yeah, I would agree. I mean, you might consider that your definitive archival format will not take this component, this overlay into account. You'll take your metadata, but you would have that as an exception of what you bring into your final definitive archiving format, because that's going to be just intermediate notes that you don't want to keep.
Okay, I would like to cut it here because we're running late. So, but I think there's many aspects to this, and we may want to organize another webinar on one specific aspect, for example, on what do you archive and what not. But we can have a look into that in the future. For now, I think I want to close the session. I want to thank Xavier and Gabriele again for this wonderful webinar, and I wish you all a very great weekend.